Good afternoon. I feel like I'm definitely coming full circle. I have a memory of some years ago, I was on a committee called the Early Admissions Committee. And the Early Admissions Committee was taking students who were bored with high school and wanted to start UVI early. And we were interviewing Antonio for early admissions. And that's when I first met him, although it was, you remember video conference? It was video conference from St. Croix. And so he started UVI as a early admission student, and I got to know him in mini classes as an undergraduate. And then 3 years ago, he started his master's program. He's been through a lot. He's been through hurricanes. He's been through beach closures because of COVID. He's been through car problems.
Oh, yes.
Huge car problems. And he's been helped by a lot of people. His committee is two people who are not at UVI. One is Sandy Wiley Echevarria, who is at University of Washington at Friday Harbor Marine Labs. And Sandy got to know Antonio when he was an undergraduate. Sandy came here for a few years, and he was a big important mentor to Antonio. And Antonio also has one of the experts, I think probably the world expert on the Halophila stipulacea invasion, who is Damien Willett from Loyola Marymount. So you may begin, Antonio.
Thank you for that wonderful introduction, Teresa. And thank you everyone for coming. This means quite a lot to me to have so many people coming to my defense. So I would like to talk about my thesis, the growth of the invasive seagrass, Halophila stipulaceae, interacting with the native Syringodium filiforme in the United States Virgin Islands. So I would like to begin by first going over what seagrass is, because I think having some background information on it is useful. So seagrass is a flowering plant, an angiosperm, with 72 different species across 4 different families. And seagrasses have the same general structure. You have a rhizome, which is buried, a horizontal stem buried beneath the sand, from which grow roots, and then from the rhizome, leaves grow and stick out through the sand. And since they are flowering plants, during sexual reproduction, they will create flowers, which give forth to fruits and seeds. Oftentimes, especially outside of academia, people will get it confused with seaweed, which is macroalgae, and they are very different. For example, macroalgae usually composed of a holdfast, which they use to grab onto large substrates, from which grows their blades. So why should we care about seagrass in the first place? Well, they're very important. They provide, first and foremost, they provide shelter and food for a number of important species. Economically important, culturally important, and even endangered. This includes sea turtles, conchs, such as the queen conch, snapper, parrotfish. Those are all very important here in the USVI. They also protect coral reefs from sedimentation disease. Mangrove, during heavy rains, sediments and runoff will occur, which ends up bringing with it lots of sediment, pollution, pathogens, and seagrass is very good at capturing those. First, they usually get captured by mangroves, and then seagrass beds act as a second line of defense before corals. They're good at capturing those things and holding onto the sediment. And it's been found that seagrass are actually have compounds that help to kill pathogens, including many that are harmful to humans. They have a high carbon storage potential. With their complex root systems, they can capture organic carbon in the water column and get sequestered beneath their roots. They also, through photosynthesis, take in carbon dioxide, which becomes part of their body, their mass. And when the leaves die, those can also get captured and buried beneath the root system, taking the carbon out of the system. Similar to on land, when you have lots of grass and other plants, those complex root systems help to keep sediment in place. And this can also be done underwater with seagrass. They have very complex root systems with their rhizomes and roots, and it helps to keep the sand in place. And then finally, they can increase the pH of the surrounding water. You see here with the dissolving inorganic carbon in the water during photosynthesis, a healthy seagrass bed, well-established one, will take in the carbon dioxide, and overall in the system, you will end up with a net loss of dissolved inorganic carbon. This causes the pH in the water to raise, which helps to mitigate the effects of ocean acidification, as opposed to when you have a less established, smaller seagrass meadow. They'll take in overall less dissolved inorganic carbon, which means there's going to be more in the system and the pH is higher. Now, this is important because corals cannot really do well in acidic waters. So if the pH of the water decreases, corals have a harder time growing their calcium carbonate bodies. So there are many native seagrasses in the USVI. Some examples of them are Thallassia testudinum, turtlegrass, Syringodium filiforme, manatee grass, Halophila shoalgrass, and Halophila decipiens, or paddlegrass. There's more than this. For example, there's also Halophila johnsonii, I believe. It's also found in the Caribbean. Now, Halophila is our early successional seagrass. They are usually the first to come in, the first to colonize, and usually the first to respawn after disturbances. They're actually very good at recovering from disturbances because when they undergo sexual reproduction, create the seeds, those tend to get buried in the sediment and they stay there. If Halophila gets removed, those seeds will germinate and new shoots can grow in. So they have a seed bank prepared. Syringodium filiforme is our middle successional, if you say. They have thicker rhizomes, thicker roots. So they come in afterwards and further stabilize the sediment, and this all paves the way to our late successional climax species of Thallassia. These usually take very long to grow, and getting started, they're a bit more at risk. But once they become established, they are actually very resilient or resistant to change. Now, Halophila decipiens is usually only found in deep waters. So you don't tend to encounter them as much, especially if you're just snorkeling. So you mostly see these three in the USVI primarily. We talked about why seagrass is important, but now we have the threats to seagrass. So worldwide, they've been declining. As of a 2008 paper, they found that 29% of the seagrass they have looked at has declined since 1879. And the speed of the decline is also increasing from 0.9% per year before 1940 to 7% per year since 1990. This is for a variety of reasons, including natural disturbances such as hurricanes. Prime example of that is here in Brewers Bay. This is a picture, an aerial photo from February before Irma and Maria. And you can see where there is seagrass. And then afterwards, we have post Hurricane Irma and Maria in November. And you can see that the seagrass has been removed from the storms. There's also the human impacts, such as boating, pollution, development. The piers built that causes a lot of stress and potential death to the seagrass in the region. Pollution also can bring with a disease and just generally will kill it. And boating, there's anchoring in seagrass beds will tear up the seagrass, but probably more well known is the effects of prop scars. As people drive through seagrass beds, the propeller, if it's low enough, will actually end up destroying the seagrass, leaving these long scars in the bed. And then there's also disease. There's seagrass wasting disease, which ends up causing the blades to turn brown and die. and now they may be facing an invasive species. So what is an invasive species? Well, NOAA describes it as an organism that causes ecological or economic harm in a new environment where it is not native. Now here in the Caribbean, we tend to have a bit of a poster child for our invasive species, and that's the lionfish, which is from the Indo-Pacific. It was first believed to have been released in Florida in 1985, and since then it is explosively spread, as you can see in this GIF here. Over the years, it has extended its range just from Florida up the East Coast and throughout the Caribbean. And it's really good at doing this because, for a number of reasons. First and foremost, there's no natural predators here eating them. There's nothing that prevents that's killing them off. On the other side, they also, when they reproduce often, and when they do, it's a lot of offspring. So combining lots of offspring fairly often with no natural predators means that they don't really have anything keeping them in check, and they can spread unabated. The problem with lionfish is that they are voracious eaters, and it's been found that if you have a lionfish on the reef, the biodiversity tends to go down. They eat pretty much everything they can to get their mouths around. Brings us to Halophila stipulacea. Now this seagrass is actually native to the Red Sea and Indian Ocean, so the east coast of Africa. And it was first found in the Mediterranean in 1894, and it's believed it came through the Suez Canal. It was quickly recognized as invasive and spread throughout the Mediterranean. And it was able to do this due to a number of factors, such as its range of conditions it can survive in and the depths it usually can be found in. This can be less than a meter, where if the minute you step into the water, you can find a bed, to over 50 meters. So that's a very impressive range of where it can survive. They're also fairly salt tolerant. They can go high salt or sometimes low salt, and they can reproduce through clonal fragmentation. So what that means is, with this one fragment, with the rhizome, the roots, and the leaves, if that were to land on the sand, a whole new seagrass bed of Halophila could grow from that fragment. And spread to the Caribbean, it has. It was originally found in 2002 in Grenada. So if you look at this map, which is from 2014, started here down in Grenada, and it has gone all the way up to St. John and over to the ABC Islands. It was found in St. Thomas and St. John in 2012. It was found in Puerto Rico in 2016, and I found it in St. Croix in 2017. 2014, it was found in Venezuela, where the first male flowers were found. In 2018, the first fruit was found, but no seeds were present. With the fruit, it raises the question if there is sexual reproduction happening. Dr. Stanford here at UVI has done some work on the genetics of the Halophila here, and they found that when they tested the Halophila, they mostly were the same genotype, which strongly suggests that everything here is from clonal fragmentation, clonal reproduction. It's the same few genotypes. So the likelihood of sexual reproduction is incredibly low. Most likely it's not happening. When talking about the spread and growth of Halophila, we can look at Dominica. In 2008, the total area was 316 hectares, of which 85% of that was Syringodium filiforme, making up 267 hectares. Halophila was only 6% with 19 hectares, while Halophila decipiens, Halophila wrighti, and Thallassia testudinum made up the remaining percentages. In 2013, though, the total area of the seagrass was 773 hectares, so nearly double what it was in 2008. Of that, Halophila is now the predominant seagrass with 86% total, making up 669 hectares, an explosive growth compared to what it was a few years ago with only 19. Syringodium, on the other hand, decreased to only 13% of the total seagrass, with 100 hectares, more than half of what it was in 2008. Halophila wrighti and Thallassia testudinum remaining relatively unchanged. Halophila decipiens, a native to Dominica, was no longer present from they were found. This was done with Steiner and Willett. So when we talk about interactions with seagrass, we need to talk about competition and facilitation, which are often used when talking about plants. Competition is when the interactions of two organisms cause harm to both individuals. And this is usually over resources. They're fighting for the resources available, such as nutrients like nitrogen, phosphorus, the space so they can actually grow and spread. Sometimes they compete for light, shading each other and preventing them from growing around them, and moisture for the water is available. On the other hand, you have facilitation, when the interactions of the two organisms either benefit both individuals or one of them is benefiting but not causing harm to the other. You often see these in nurse plants, which are sometimes found in more extreme environments like deserts. They provide benefits to other plants that grow near them, such as stabilizing the sediments, making it easier for them to grow, protecting them from environmental hazards or conditions such as strong winds or currents, increasing the moisture in the area if it's a very dry area, or even shading the plant if the other plant can't really tolerate high levels of light. So we can talk a lot about competition and facilitation, how the plants interact, but you also need to think about how the seagrass is being impacted by nature. And so we have hurricane impacts, which are known to be damaging from seagrass beds, as we said earlier, with Brewers Bay and Maria. Following the passing of Hurricane Hugo in Puerto Rico, they found that 3 square kilometers of seagrass is either removed or buried beneath the sand. And after Hurricane Maria, they found a significant loss of seagrass in many parts of Puerto Rico. And this is important because the USVI is in the path of many storms. Within the last 3 years, we've suffered from 2 Category 5 hurricanes, Category 1, and 2 tropical storms. And there is projected that's going to be many more storms in the future, and they're going to be stronger. And this can provide an opportunity for faster growing seagrasses, such as Halophila stipulacea, to take root. Going forward, if I say just Halophila, I'm referring to Halophila stipulaceae, unless I specify otherwise. So when we talk about the storm impacts, we need to talk about the resistance and resilience. Resistance is defined as how well a system remains unchanged after a disturbance, while resilience is how well a system recovers after a disturbance. So for the sake of seagrass, you have a high density bed, and if you have high resistance, after disturbance occurs, a hurricane, you remain relatively unchanged. On the other hand, if you were to have low resistance but high resilience, after the storm, you wouldn't see a severe decline, but you would quickly recover to similar conditions from pre-storm. Low resistance and no resilience, you'll see your density drop and you won't recover very quickly if you recover at all. And then you have opportunistic, where you might not be doing well before the disturbance, but after the disturbance happens and those that are currently dominating the region are removed, you then have an opportunity to actually increase and spread and thrive. Which leads us to one of the problems. There's a lot of unknowns involving Halophila in the Caribbean. What are the interactions with native seagrasses? Are they competing with one another? Is Halophila displacing our natives? Are they facilitating each other's growth? Or are there no interactions and they're not really interacting too much or doing their own thing? What about the interactions with native fauna? Are native animals eating it? Do they eat it if they have to, but they prefer our natives? Do they not care either way? Do they use it as a habitat? Are they spending time above it? Do they avoid it? Is it the same fish swimming above Halophila that you would normally find with Syringodium or Thallassia, or is it different types? And is there any benefits to having Halophila here? We've talked about how important seagrass is and all the benefits they provide. This seagrass is quick to spread over any sandy area, which might not currently be utilized by our native seagrasses. Is it better to have it there than have nothing at all? Can Halophila actually provide a similar service to our native seagrasses? So I wanted to look at the first one, which is the interactions between Halophila and our native Syringodium filiforme. So the questions I wanted to ask or answer was, how does growing together compare to growing alone? To answer that, I did a common garden experiment with them. What does the donor beds look like from where I took my samples? And so that I did a monitoring experiment during the same time period as the common garden. And how well do they respond to storm impacts, which again was another monitoring. This came about due to the fact that Tropical Storm Isaias passed through here during the middle of my experiment. In previous attempts and pilot study, I realized that storms can severely impact the plots. So I took data right before the storm just in case and decided to add a storm impacts aspect to this. So these are my hypotheses that I wanted to test. Is Halophila negatively affected by Syringodium? Is there competition occurring? Is Halophila not affected by Syringodium? No interaction. Or is Halophila being positively affected by Syringodium? There's facilitation occurring. I want to see the same thing for Syringodium. Is there competition, facilitation, or interaction? So I did my experiments in Lindbergh Bay here in St. Thomas. I conducted it in the swim zone, which is a largely unvegetated area. There is some Halophila and Haloduli growing in the area, but for the most part it is mostly sand. This way I would not have to remove an area of seagrass to put my stuff down. The other reason I chose the swim area is because you are not allowed to anchor within the swim area. And there are occasionally boats that will anchor here in Lindbergh. It's far enough offshore, though, and in deep enough water that most beachgoers do not go out that far. They don't really go out where the plots were. So I was not too worried about human interactions affecting it. This was done at a depth of 2.5 meters. The Halophila beds that I pulled from, the large monotypic beds, is located here at 3 meters in depth. And the Syringodium beds were located over on the western end at 3.5 meters in depth. The reason I chose the Syringodium bed there is because I was looking for large monotypic seagrass beds, and finding Syringodium that fit that criteria was difficult, and this was the only bed that I found that fit that criteria. There is another monotypic Syringodium in shallower waters, but it was not big enough for my purposes. As I looked through the bay, I made note that there's Thallassia growing, Thallassia testunum, Syringodium filiforme, Haloduli wrighti, and Halophila stipulaceae. And this has been there for some time, even before Maria. These have been found there and is documented. So for the common garden experiment, the transplant experiment, this took place over 2 months from June 6th to July 28th. There were 15 plots that were sampled bi-weekly. I did a pilot study, and I found that after the 3rd month, there was quite a bit of growth, and we couldn't really capture to see what was going on with the monthly sampling that we were doing before, and we felt that bi-weekly would work better, give us a good result. There are 3 treatments in a randomized block design, where you have Halophila growing by itself, Syringodium growing by itself, and Halophila and Syringodium growing together. Each sample, the plots are 1 meter square and were spread across a depth of 2.5 meters. Each sample was 15 centimeters in diameter when I took them, and they were positioned 10 centimeters apart when grown together. I chose 10 centimeters because I wanted them to be close enough that they could interact with one another without being forced to by being placed right next to each other. Shoot density was measured by counting the total number of shoots in the plot, while percent cover was visually estimated using a 1 meter square quadrant. Max shoot height was measured with a ruler to the closest millimeter. For my statistical analyses, I used a Shapiro-Wilk test to check for the normality of the data. And if I met the parameters for a parametric test, I did a t-test. And if not, I used a Mann-Whitney U-test. So on my x-axis here, you have the dates of the observations. And on the y-axis, you have the shoot density, the number of shoots per meter squared. Orange is representing plants that were, samples that were grown alone, while green is those that were grown together. Circles represent Halophila, while triangles represent Syringodium. So when you look at Halophila growing alone and Halophila growing together, I did a t-test at the end of the 2-month experiment period, and I found that there was a significant difference between the two of them, that Halophila growing alongside Syringodium did significantly better than by itself. When I look at Syringodium growing alone and growing together, the t-test showed that there was not a significant difference between the two of them. Now on the graph, it may show otherwise, but I believe that may be, they show that there's a significant difference, but I believe that's due to having low statistical power due to only having 5 replicates. For percent cover of the meter squared, Halophila growing by itself and Halophila growing together came back with a t-test result at the end of the 2 months that there was a significant difference, that the percent cover was significantly greater growing with Syringodium than by itself. Syringodium, once again, did not have a significant difference between the two of them. In terms of max shoot height in millimeters, for both Halophila and for Syringodium, the t-test showed that there was not a significant difference between those grown alone or those grown together. And it's important to note the differences in height. Halophila is a very short plant, 3 to 5 centimeters on average when I was the average when I was doing those. While Syringodium can be upwards of 20 centimeters, or usually between 10 to 20 was around where I mostly found them. So they usually get to Halophila tends to get to that max height fairly quickly, so you don't see much variation with them. So my conclusions for the common garden was that at the end of the 2-month period, Halophila shoot densities and percent cover were significantly higher when growing alongside Syringodium, which means that Syringodium is likely facilitating the growth of Halophila. Now for the donor bed monitoring, this took place from June 6th to August 1st over the 2 months. These were sampled monthly due to the pilot study showing that my results from the pilot showed that there wasn't much change, so we felt that monthly sampling would be fine. At each bed, 10 observations were made, and this was done by taking a 1 meter square quadrat and placing it haphazardly. Percent cover was then visually estimated using the quadrat. And in the center, a 100 centimeter square quadrat was placed, and shoot density was measured in it and converted to meters squared. The max shoot height was collected using a ruler to measure the tallest blade to the nearest millimeter in the center of the 100 centimeter square quadrat. For my statistical analysis, I did a Levene's test to test for the normality. And if it met the parameters for a parametric test, I did a one-way ANOVA. And if it did not, a Kruskal-Wallis test. On the x-axis is the dates of observations, the 6th of June, 4th of July, 1st of August. And on the shoot density on the y-axis, Halophila is orange and Syringodium is green. And this shows when Tropical Storm Isaias passed through the area. And for both Syringodium and for Halophila, a one-way ANOVA showed that there was not a significant difference over the 2-month period. For percent cover, for Halophila, we see that the one-way ANOVA, there was a significant difference across the dates, and a 2-key showed that it was between the 6th of June and the 4th of July, there was a significant increase in the percent cover of Halophila. For Syringodium, the Kruskal-Wallis revealed that there's a significant difference, and a Dunn's test showed that there was a significant difference between the 6th of June and the 1st of August, so the beginning and the end. And finally, for max shoot height, once again, for both species, there was not a significant difference for them. So my conclusion for this was that the percent cover of Halophila and Syringodium changed between observations. The changes could be due to seasonality, as the summer is when these seagrasses tend to be the most productive and grow the most. But there might be impacts from passing of Tropical Storm Isaias, because I did see that the Halophila cover did decrease. For storm resistance and resilience, the data was collected on July 28th, which was 2 days before the storm. The storm occurred on July 30th. Then August 1st, I collected data afterwards for the post, and August 22nd, 3 weeks after the passing of the storm. July 28th to August 1st was used to measure resistance, while August 1st to August 22nd was used to measure resilience. These were the same plots as the transplant experiment, and the reason why the transplant experiment didn't continue, because after the passing of Tropical Storm Isaias, some plots were damaged and some were completely wiped out. So by using the chain plots as a transplant experiment, I can also test whether the presence of the other species affected resistance or resilience. Shoot density was measured by counting the total number of shoots in the plot, and percent cover was visually estimated with a 1 meter square quadrat, same as before. And the max shoot height was also measured with a ruler to the closest millimeter. For my statistical analysis, I did Levine's test to check for the normality. And since all of them met the parameters for a parametric test, I used a two-way ANOVA. Here on the x-axis, you have the three observations. So the 28th of July, pre, 1st of August, post, and 21, 3 weeks after. And for Halophila, growing alone and together, or this one, a two-way ANOVA showed that there was not a significant difference between date, treatment, or the interaction of date and treatment. For Syringodium, however, growing alone and growing together, we see that there is a significant difference in treatment, but not date or the interaction of date and treatment, which means that Tropical Storm Isaias did not have an impact on shoot density. This rather shows, but from this we can infer that Syringodium growing alone did significantly better than together. For percent cover, Halophila growing alone and together, the two-way ANOVA showed that there is a significant difference in date, but not treatment, in the interaction of date and treatment. And the two keys showed that these were between the 29th of July, or 28th of July, I missed that. So pre and post. So between the resistance, there was a significant difference. It significantly decreased. And then we also see it again for the 28th of July and the 21st of August. Those were also significantly different. For Syringodium, alone and together, we see that there is a significant difference in date as well, but not treatment or interaction of date and treatment. But the two key could not determine which months those were. So we do know that the storm did have an impact on Syringodium percent cover, but we're not sure when. And once again, with the max height, we see that there is not a significant difference between either species across date, treatment, or interaction of date and treatment. So my conclusions for this is that Tropical Storm Isaias decreased the percent cover of both Halophila and Syringodium. And this means that even relatively weak storms can impact small patches of seagrass. So to recap, Syringodium filiforme may be facilitating the growth of Halophila stipulacea. Halophila shoot density and percent cover were not significantly greater when growing alongside Syringodium while not having a negative impact on it. The donor bed seemed very stable, and changes could be a result of seasonality or storms. The donor beds of Halophila and Syringodium did not see much difference in shoot density or height, but there were increases in percent cover between some observations, or significant changes in percent cover between some observations. Both Halophila and Syringodium were affected by storm damage. Halophila and Syringodium had lower percent cover after the passing of Tropical Storm Isaias. So what does this all mean? Well, there is evidence of facilitation occurring between Halophila and Syringodium. Kelsey Sakido also presented some result at ASLO on her experiment of growing Halophila with Syringodium in a mesocosm. Her results showed that there were, when Halophila was growing alongside Syringodium, it did better, but she found that when Syringodium grew better by itself. Looking back at the storm impacts, we did see that Syringodium growing by itself did do better than together with Syringodium. This might be due to the length of my experiment not showing what would happen long term. Halophila may have an easier time spreading through the Caribbean if Syringodium is facilitating its growth. Syringodium is quite common in the Caribbean, which means that if it is helping Halophila grow, it might have an easier time spreading. And storms are able to damage both beds, which may provide opportunities for Halophila to spread faster. As we saw, that storms can actually remove seagrass and open up substrate for them to grow upon. So what are the possible mechanisms for the facilitation? I'm not entirely sure. It could be chemical, where with the nutrients, nitrogen and phosphorus, maybe they're utilizing it differently or it's helping one another with it. Could be stabilization of the sediment. Syringodium rhizomes are much thicker and they stabilize the sediment better. So maybe that's helping the Halophila grow. Or, which is my best guess right now, is that that Syringodium is protecting Halophila from strong currents, which might rip it up or impede it. As we said earlier, Syringodium is much taller than Halophila, so it could help protect it. The limitations of my experiments were that the length of experiment, first and foremost. Originally, it was meant to be 3 months, but was cut short due to Tropical Storm Isaias. So going for a longer period of time could get a better understanding of what is happening between the interactions of these two seagrasses, as well as what the long-term effects are. So extending the length would be great. The location of the experimental plots is a largely unvegetated spot, so it's possible they would not grow there naturally. There is Halophila and Syringodium growing around that. It was just sparse, and over time I have seen Halophila start encroaching from the swim area. And the seagrass beds are far apart from each other in slightly different depths, where I got my samples from. So it's possible that the differences in the environment, such as what was the sediment composition, the chemicals in there, or the nutrients in there, the grain size is different, depth, maybe light, those could all be factors that could be influencing these. So for further work, I think additional replicates would be fantastic. Doing more would give a better understanding. It would increase the statistical power. A longer duration would be probably one of the best things, as it would give a much better idea of what's happening long term. And if long enough, it could actually also look into the seasonality differences. Using additional bays would also be helpful, because this might be localized to just Lindbergh that we're seeing things. But if we see in other bays, it will be helpful. Nutrient sampling to see what the nutrients in the ground are. The grain size analysis impacts herbivores are having on these seagrasses, as well as algae. And including Thalassia testing in Halophila ready would also be fantastic. Thalassia is hard to transplant, so that one might be a little bit more difficult. But Halophila would be very good to look at, considering it's an early successional species as well. So I have a quick clip from my data collection. This was by the Syringodium beds. I would collect the core sample, put it into the bin, and it was too heavy to actually swim with, so I ended up taking my fins off, clipping it to my VC, and then running across the seafloor with the bin, my partner close behind. So while not only did it get my my thesis completed, it also fulfilled a lifelong goal of mine to walk across the seafloor, which is something I've wanted to do since I was a child. This is always difficult. Go. So for thanks, I would like to thank Kyle Jarris so much for his help throughout the entirety of this study. I would not be here without his help. He's been there since the beginning when I was planning this, field work, writing my paper, presentation. He's helped me throughout the entire process, giving me, bouncing ideas off each other, helping me. I cannot thank you enough. Joseph Townsend, he helped me a couple times with field work, including with the pilot studies. My cohort, the 2017 HURA cohort, for the help and support getting through the start of the program, because 2 Category 5s at the start is definitely an interesting way to start my grad school. Lennox, Rike, Tachi, Lux, Drac, and Becca for helping keep my sanity. Numerous other friends and family that it would take forever to list. And my funding, I'd like to thank the iOS Karakus Okovi Bamboo Sumerian Ward, as well as the Lana Vento Fund. Thank you, and any questions?
I am in
I have a question.
Yes.
Were you in any way able to take into account, or did you see any evidence of turtle grazing in your plots?
When I was looking at the plots, we didn't see any obvious signs of grazing. From what I could tell, if I saw something, I tried to make note of it, but I didn't really notice anything.
Antonio, I have maybe kind of a follow-up on that. Did you notice by the end of the experiment if there were like any things like fish associated or anything that you saw swimming around there that was bare beforehand that you're now seeing things associate with it?
When I went to go look to collect data, often didn't see much fish swimming around the areas. I did notice some fish with some sunken beach chairs. But when I actually went out to collect data, fish were not very prevalent near the plots.
Huh. Interesting.
I have a
Antonio,
question. Oh, go ahead, Steve.
This is Steve Ratchford. I noticed that one of your early slides about data from other places showed that other seagrasses were reduced in areas where there's Halophila. And that, immediately would make me think there might be some competition going on here. Do you have any idea why you didn't see sort of a reduction due to the Halophila? Seemed like you were saying that the Syringodium was fairly unimpacted.
That's correct. I'm not entirely sure why I didn't see some results. My best guess would be because the length. Maybe it might take longer before such things will be seen. During the pilot study, when I did a trial run of this, I found that by the third month, the end of the third month, was when I started seeing significant increase in growth. So it could have been then. So maybe getting cut short, it's not allowing me to see the full picture.
As just a quick follow-up, did your plots where you had your two species, the Syringodium and the Halophila, was there enough time for them to grow together to go across that 10-centimeter distance?
Syringodium was very slow to spread outward.
Yeah.
Though I did see some Halophila, which occasionally you could see some, I want to say tendrils, start heading toward the Syringodium in almost all directions. So I do believe that Halophila was capable of heading toward the Syringodium. In some cases, it did get close. I believe we saw some interaction, but not much.
Antonio, I had a question. This is Kristen.
Yes.
I was curious about epiphyte loads and if those were different among your different treatments in the garden experiment, or if that was something that you had quantified at all.
I did not quantify epiphyte load, though I do know that I've seen quite a bit of macroalgae growing in the region, because in each plot there are two rebar pieces that were in opposite corners to help for placing the quadrat and marking the plots. And those tended to build up quite a lot of macroalgae. There was some epiphytes on both the Syringodium and the Halophila, but I don't have a number, a quantity of it.
it would be interesting to think about maybe the role of those in trying to explain the growth patterns that you observed too. So that might be something to think about.
Most definitely. I think looking at the algae impact on the species would be something fantastic to look at. There was some other, like I said, macroalgae growing near the plots and in the plots, but not much of it. And some of that may have been brought with the transplants.
I had one other question for you too. Was it looked like in the garden experiment that there was like a delayed effect? And so I'm curious to what you think about whether or not you would expect that if you had run your experiment for a little bit longer, if you would have expect those differences to continue to grow on some of the metrics, like I think you had sheet density and percent cover where those were persistent.
I would expect that. I think there was a delayed effect because I saw something similar with a pilot done back in the winter. The first like month or two, there wasn't really much. We were actually sitting there talking like there's not much going on. Third month, we were joking, we're not going to see anything else. And on the third month, we actually saw quite a bit increase. There were some problems with that study though, so I ended up redoing it now.
Mm-hmm. Remind me of the dates on your common garden experiment though. Did that end before Isaiah's hit?
Yes, so it started on June 6th and ran to July 28th. It was originally meant to go until around August 28th or so. Mm-hmm. But from last year, I remember trying to get some plots, which then ended up getting destroyed by Tropical Storm Karen and Hurricane Dorian. Those ended up destroying my plots. So I remember storms tended to not do well with my plots, did not do well with the storms. So I ended up ending the experiment on the 28th. I took data then just in case. And when I looked on the first, I noted, or after the storm, I noticed that some of my plots were damaged. Some of them were actually completely wiped out. So I ended the common garden experiment on the 28th and decided to add a hurricane or tropical storm impact study onto this as well.
So it would be kind of interesting to combine those. Sorry, Edwin, just one last follow up there, and then I'll let you jump on there. It would be interesting to combine those though, because that would be, I don't know, that would be a really interesting question about, well, what happens to your to your garden experiments, you know, like does it reset for, and is that species specific, or does it change if there's a species interaction? It's really interesting to think about.
I had a mix of plot removal. Some Halophila were ripped out, some Syringodium were ripped out. Sometimes it was those growing together, sometimes it was those growing alone. Mm-hmm. And I just remember looking at my project like, well, that's dead.
Okay, thanks. Go ahead, Edwin.
Yeah, you mentioned that you saw Haladul in that area too, where, I mean, I know it wasn't part of your experiment, but I'm kind of curious whether it was mixed in or whether it was like segregated somewhere else in that area.
Let me go to my map. Okay. So there are some fairly good sized Halophila beds scattered throughout here, and then over by the actual experimental site, you'll have small patches here and there. Sometimes you'll only have one or two shoots sticking out. Sometimes you'll have a lot more. And it's more or less scattered throughout the deeper area over here, and sometimes into the shallows over on this end.
Now, if you look at that same map to the left, if you move the cursor, there seems to be like, yeah, right there, those seem to be like really shallow. Is that Halophila or is that Thalassia?
That is a mix of Thalassia and Halophila, and as well as some Syringodium.
Okay, thank you. I think that on behalf of the island, we don't want you to run any more experiments because every time you do that, there's a hurricane.
Please don't remind me.
Yeah. So please, your next experiment, run it somewhere else.
Sounds good. I like that idea. Let's go somewhere else where there's no hurricanes.
Yeah. Nice job.
Good job, Antonio.
Thank
Yeah,
you.
nice job.
Antonio, this is Marsha. Could you explain again, how did you calculate percent cover?
Yes, so I took a 1 meter square quadrat. And so if this would cooperate with me. Okay. So this was the plots with the sample planted in the center. And on each corner, there was a piece of rebar, which I used to mark the plot. And also I could slot, basically just plop down the quadrat with the rebar in either corner. And I would visually estimate that and write that down in the field.
So it was a visual estimate rather than
Yes.
taking a picture and digitizing.
Yes,
Thank you.
I had problems with that in the pilot study with camera work. So I decided to simplify it and just do visual estimations in the field.
Antonio,
Yes.
so did I understand that you then have determined that you believe the Syringodium is facilitating the Halophila growth?
Based on my
Yes.
current results, yes.
And that you suggested that perhaps this was related to the height of the Syringodium reducing current?
Possibly. I do not know for certain.
So how might you
That's
test
just my
that?
best guess. I do know that there's been some work done where they can test the rate of flow in seagrass beds. So I can see what the rate of flow is above Halophila and above Syringodium, and then maybe test the rate of flow of Halophila near Syringodium to see if maybe it's different.
I don't know how valid it is, but we also tried trimming. So we had beds of, we were looking at Thalassia a few decades ago, and we tried trimming some of them to see if that could, simulate the shorter growth of Thalassia. But I thought you might have some ideas too about what else you might do in perhaps a lab setting. But I don't know for sure that the trimming would help you, but it seems like an interesting question to answer.
Yeah, understanding the mechanism of the facilitation, I think, would be something important to look at and try to figure out.
Well, and sort of first to eliminate some of your ideas about what might be going on is helpful, so as to narrow your focus, because I expect you may be looking at seagrasses with interest in the future, yes?
Yeah, I would like to continue with seagrass. I think at this point in time, my interests lie in coastal ecosystems, so mangrove, seagrass, coral reef, especially the interactions between the three. That seems to capture a lot of my interest right now.
Thank you.
Hey, Antonio, Paul Jobson. So I had to step away for a little bit, so you may have covered this in your presentation or during one of the questions. So did you look into the effects of sort of enemy release, or the fact that the Halophila might have less predators, such as turtles or invertebrates eating it?
I haven't looked into any herbivory. I think I did see if I tried to make note of it, if I saw any examples of it, but I didn't really see much. That's something I didn't make note of, like if I saw it, but I didn't really see much evidence.
Yeah, there was some work by John Cassell, who graduated in 2018, showing that the green sea turtles were foraging more over mixed beds and Syringodium beds than the monotypic beds. So, but anyways, that might be releasing the Halophila from a predator. So.
Paul, do you know off the top of your head how common sea turtles are in Lindbergh?
On the top of my head, I haven't been there recently, but we used to survey there and see green sea turtles every time we survey. Not as in Brewers,
Mhm.
but they were there. So that was before Hurricane Irma, though, so it may have changed since then.
Paul, that bay was omitted from Jessica Michael's study, correct? We could potentially look at that data if that was chosen.
Right. Yeah, Jessica, that was not one of the bays that Jessica chose. I mean, she did Druff Bay, which is sort of across the waterway there on Water Island. And that was actually one of the highest density green sea turtle bays. And of course, Brewers was her second highest green sea turtle bay. So I would imagine they're there. I just don't think anybody's done that, been counting there. At least nobody in my group has.
Hey, Antonio. Nice to talk. I guess one question that I have is, why did you choose filiforme instead of like one of the other seagrasses, knowing that it's the middle colonizer and Halophila has been looked at as a possible competitor with turtlegrass in terms of being a late colonizer and an early colonizer as well? Is it because it was the middle ground? Sorry. Loaded question.
Yeah, a little bit loaded. So the reason I chose Syringodium was I had already had it in my mind I would do Syringodium and Halophila. And I felt I would be able to find Syringodium. I'd also, when I was first thinking about doing my project, I had actually thought about doing it on St. Croix. I ended up deciding to do it here on St. Thomas instead. And especially early on, as I was getting more and more familiar with seagrass, I realized why, like, I thought, I'd be able to find Syringodium quite often. It should be quite easy to find monotypic bed. And I found that later on it was harder to find a monotypic bed of Syringodium. But at that point, I was already committed to doing Syringodium Halophila. And that's what I decided on and decided to stick with it. And I may or may not have had trouble differentiating between Holodule and Syringodium at the beginning of my program. That's my big shame there.
At least I know one of the bays that I'm going to be looking at has all the different types of seagrass in it.
Okay, so if there's no more questions from the audience can all leave, and Antonio and the committee can do the